The overall goal of this procedure is to separate and compare the monosome and polyribosome fractions from cell lysates. This is accomplished by first culturing the cells of interest. The second step of the procedure is to prepare the cell lysate. The third step of the procedure is to centrifuge the lysate through a sucrose gradient. The final step of the procedure is to fractionate the gradient and visualize the different ribosomal populations through a UV trace. Ultimately, results can be obtained that show changes in translation under different experimental conditions through polyribosome profile analysis.
Hi, I'm Anthony Esposito from the lab of Dr. Terri Kinzy in the Molecular Genetics, Microbiology, and Immunology Department at UMD&J Robert Wood Johnson Medical School.
And I'm Marcus Lewis, also from the Kinzy lab here at RWJMS.
Today we're going to show you a procedure for polyribosome analysis.
We use this procedure in our lab to study alterations in protein synthesis that arise from mutations or environmental factors.
So let's get started.
Sucrose gradients should be prepared 1 day before use to allow gradients to become continuous. Mix the appropriate volumes of 7% and 47% sucrose stock solutions to make 48 milliliters of each of the following sucrose concentrations: 7%, 17%, 27%, 37%, and 47%. Add 1 molar DTT to each sucrose solution to a final concentration of 1 millimolar. In order to pour the gradients, attach a Pasteur pipette to a 20 milliliter syringe by securing and sealing with parafilm.